Hi viewers, in this class we are going to discuss the agglutination antigen antibody reactions. Apart from the antigen antibody reactions, we are going to discuss the specific reaction which is known as the agglutination. Under this topic we will talk about introduction to the topic, mechanism of agglutination, prozone, titer and the zeta potential, and various common agglutination tests and the applications of agglutination reaction. The reactions of antibody with a multivalent antigen, that is a particulate antigen, means insoluble particle, that results in the cross-linking of the various antigen particles by the antibodies. This cross-linking eventually results in the clumping or agglutination of the antigen particles by the antibodies. Here, for the first time, two bacteriologists, namely Herbert Durham and Max von Gruber, both of them discovered specific agglutination in the year 1896. Hence, this is known as Gruber-Durham reaction. Gruber-Durham reaction. For the first time, Gruber introduced the term agglutinin from the Latin for any substance that caused agglutination of cells. Later on, French physician Fernand Weidel put Gruber and Durham's discovery to practical use later in the 1896, using the reaction as the basis for a test for typhoid fever. Weidel found that blood serum from a typhoid carrier caused a culture of typhoid bacteria to clump, whereas serum from a typhoid-free person did not. This Weidel test was the first example of serum diagnosis. Later on, Austrian physician Karl Landsteiner found another important practical application of the agglutination reaction in the year 1900. Landsteiner's agglutination tests and his discovery of A, B, O blood groups was the start of the science of blood transfusion and serology, which has made transfusion possible and safer. When the antibody interacts with its particulate antigen, it results in agglutination and the antibody is known as agglutinin. Antibody is known as agglutinin and the antigen is known as agglutinogen, and it occurs in equivalence zone. In general, IgM is known as anti-agglutinin, whereas IgG is incomplete antibody causes partial agglutination. Finally, agglutination is defined as the formation of clumps of cells or inert particles by specific antibodies to surface antigenic components or to antigenic components absorbed or chemically coupled to red cells or inert particles, that is known as agglutination. Here you can observe titer. The agglutination of an antigen as a result of cross-linking by antibodies is dependent on the correct proportions of antigen to antibody. These agglutination tests for antibodies to the bacterium Brucella abortus present in the serum of an infected individual. How we will do that? Some test tubes are taken containing twofold dilution, serial dilution of the serum, ranging from here various examples are there. Here 1:20 to 1:1280. So that means a number of test tubes were taken and serum is added ranging from 1 is to 20 to 1 is to 1280. To all these, equal amounts of a suspension of Bacillus brucell abortus added. The result of the test indicates that agglutination occurs at the dilution of serum of 1 is to 80. It starts from 1 is to 80 to 1280. There is no agglutination in the dilutions like 1 is to 20 and 1 is to 40. That is, the highest dilution of the serum that still causes agglutination but beyond which no agglutination occurs is termed as the titer. So here it is 1 is to 1280. See the same another example for this. Here we can easily explain that. Here also there is a serial dilution method where it starts from 1 is to 5 to 1 is to 5120. So here there is no agglutination in these two test tubes, and here also there is no agglutination at all. In between that there is agglutination in between 1 is to 20 to 1 is to 1280. So here there is no agglutination at all. This is known as a prozone, and beyond the titer volume, titer is 1280. How we will define the titer? The highest dilution of serum that still causes agglutination but beyond which no agglutination occurs. After that no agglutination takes place. So that is known as the titer volume. Here the titer is 1280 is the titer. See here we can also define the prozone. The tubes with high concentration of serum where agglutination does not occur, that represent a prozone. In the prozone, antibodies are present in excess amount. Agglutination may not occur at high ratio of antibody to antigen because every epitope on one particle may bind only to a single antibody molecule, preventing cross-linking between the different particles. So, where due to the more amount of the serum, less amount of the antigens, no agglutination takes place, that zone is known as the prozone. In the another example, you can see the titer is 1 is to 80. 1 is to 80 is the titer. Up to 1 is to 20, 1 is to 40, 1 is to 80 only agglutination takes place. After that no agglutination. So this is the highest level where actually the agglutination takes place. Beyond that no agglutination. So this is about the titer. And one more thing you have to observe, zeta potential. Zeta potential can be explained as the surface of certain particulate antigens may possess an electrical charge. For example, the net negative charge on the surface of RBC, red blood corpuscles, caused by the presence of sialic acid. When such charged particles are suspended in saline solution, an electrical potential called the zeta potential is created between particles, preventing them from getting very close to each other. This introduces a difficulty in agglutinating charged particles by the antibodies. Some of the times it may also happen. Here in the diagram, type A blood donor and type B antibody in type A blood recipient. So there is no agglutination at all. Whereas type A blood donor and type A antibody in the type B blood recipient, that is donor is from A group and recipient from B group. So due to the presence of the anti-A antibodies in the recipient, there will be a agglutination reaction in the recipient. The same thing. And come to the various agglutination tests which are commonly used nowadays, namely slide agglutination. Under this, latex agglutination test for antigen detection, latex agglutination test for antibody detection. Second type is the tube agglutination, which is explained by the Vidal test. Cold agglutination, explained by the Weil-Felix test, Paul-Bunnell test, and passive agglutination, which is explained by the HEMAGGLUTINATION test. These are the tests which are commonly used for various detections of antigen and antibody in the agglutination reactions. See the slide agglutination test. This test is designed to observe the direct agglutination reaction of known antibody and unknown particle antigen on slide. This reaction is simple and rapid. Usually it is used to detect qualitatively the antigen or antibody such as bacteria, identify and ABO blood type assays. They comes under this type of test. And here you can see in the slide agglutination test, there is a latex agglutination test for agent detection, antigen detection, and latex agglutination test for antibody detection. They comes under the slide agglutination test. The latex agglutination test for antigen detection is used to determine the presence of capsular antigen of Haemophilus influenzae and Streptococcus species, and latex agglutination test for antibody detection used to detect cytomegalovirus, rubella virus, varicella zoster virus, heterophile antibody of infectious mononucleosis. So anti-streptococcal antibodies, mycoplasma antibodies can be detected by the latex agglutination test for antibody detection. And coming to the tube agglutination test, it is also known as the standard agglutination test or serum agglutination test (SAT). Test serum is diluted in a series of tubes, double dilution method. Constant defined amount of antigen is then added to each tube and tubes incubated at for 20 hours at 37 degrees Celsius. Particular antigen clumps at the bottom of the test tube. Test is read at the 50% of agglutination. It is a quantitative method. Confirmatory test for ELISA reactors, all these comes under the tube agglutination. The best example for the tube agglutination are the screening test for the brucellosis, and as well as the Vidal testing. And coming to the cold agglutination, so it is based upon the temperature. The patient sera agglutinated human O group erythrocytes at 4 degrees Celsius. The agglutination being reversible at 37 degrees Celsius. So it is based upon the temperature. Then coming to the passive agglutination, an agglutination reaction that employs particles that are coated with antigens not normally found in the cell surfaces. So the particle carriers include RBC, polystyrene latex, bentonite, and the charcoal. These are used as the carrier molecules. And hemagglutination that detects the antibody to erythrocyte antigens. If sufficient concentration of antibody is present, that leads to the antibody cross-linking and results in the agglutination. If it is non-reactive or antibodies are insufficient and there is no agglutination takes place. Here, binding different antigens on the RBC surface is equal to that detects the antibodies to antigen other than those present in the cells. It is a hemagglutination. See here, we will see the ABO blood groups. Here the sample is blood group A. How they react with the addition of the anti-A or anti-B antibodies. We have taken A blood group individual's blood and we have added anti-A antibodies and anti-B antibodies to both the test tubes. So due to the presence of the antigen A in the blood group A people, so that here agglutination takes place and here no agglutination. If it is a B group, it clumped with the antigen B clumped with the anti-B antibodies. So here there is no agglutination. And if it is blood group O, due to the absence of lack of antigens, there is no agglutination in both the tubes. Whereas if it is blood of the AB group individual, due to the presence of both the antibodies, so there is agglutination in both the tubes. So finally, the direct agglutination and the indirect agglutination. When the antigen is in the natural constituent of a particle, the agglutination reaction is called as the direct agglutination, where the particulate antigen that interact with the corresponding antibody and form the agglutination reaction, agglomerate otherwise. When the agglutination reaction takes place between antibodies and soluble antigens and the antibodies that had been attached to an insoluble particle, the reaction is called as the indirect agglutination or passive agglutination. See here, if you see the difference in between these two, here it is a particulate antigen, but here it is a soluble antigen. Particulate antigen directly bound to the corresponding antibody and forms the visible agglomerate, whereas the soluble antigen comes in contact with the sensitized particulate particles, and both of them underwent the reaction with the corresponding antibody and forms the visible agglomerate. So it is indirect agglutination reaction, whereas this is the direct and this is indirect agglutination reactions. So the agglutination reaction is widely applicable and clinically. Major applications include generally erythrocyte typing in blood banks, diagnosis of various immunologically mediated hemolytic diseases, drug-induced autolytic anemia, test for rheumatoid factor, and confirmation test for syphilis, latex test for pregnancy, which involves the detection of HCG, human gonadotropin, in the urine of the pregnant women. So here there are different applications of the agglutination reaction. Slide agglutination, identification of cultures like Salmonella, Bordetella pertussis, typing of pneumococci and streptococci, and blood grouping. Whereas the tube agglutination that applied for serological diagnosis of enteric fever through Vidal, brucellosis, typhus fever through the Weil-Felix reaction, blood grouping, Coombs test, all these under the applications of the agglutination reaction. So thereby we have concluding this topic by knowing what is agglutination and what are the different tests which are used for the agglutination. Thank you one and all.